Okay, welcome everybody to our next snack seminar. Today I'd really like to welcome Adeel Razi. So Adeel is from Monash, and we have a really good connection because my former PhD student, Leonardo Novelli, went down to Monash to work with Adeel as a postdoc. So as those of you who have been following Snacks know, Leo spoke to us earlier in the year, not one that we recorded and posted to YouTube, but spoke to us earlier in the year about some of the joint work he's been doing with Adeel. And today it's a pleasure to have Adeel give us more of the bigger picture, I guess, of what's going on down there. And it'll be really interesting to find out about that and to find out a lot more about dynamic causal modeling, because that's not something that I've done any work in myself, but have been interested to find out a lot more about. So formally, as I say, I'd like to welcome Dr. Adeel Razi. He's an associate professor at the Turner Institute for Brain and Mental Health in the School of Psychological Sciences at Monash University. He's the head of the computational neuroscience lab there. His research is cross-disciplinary, which, again, those of you following Snacks know that we love. It combines engineering, physics, and machine learning approaches to model complex multiscale network dynamics of brain structure and function using neuroimaging. He's currently an NIH-MRC investigator on the Emerging Leadership Program, a CIFAR Atsurelli Global Scholar in their Brain, Mind and Consciousness Program, and was an ASA DECRA Fellow up until this year. He has undergrad degrees in electrical engineering, Masters in Communications Engineering, and a PhD in electrical engineering. So Adeel, as I say, it's a great pleasure to welcome you. And we'll throw over to you for the talk.
Thank you. Thanks, Joe. Yeah, it's a great pleasure to be here. Lots of familiar faces here. So, so we'll be talking about some of the work that we have been doing over there. And yeah, okay, so let me start with so we'll be talking about generative models. So that would be the main theme here. We will be going through methods in the beginning, and then I'll show an application of those in psychedelics. Okay, so what do we do? So what we do is, as Joe was saying, do multidisciplinary research. So we use brain recordings from various multiple imaging modalities, MRI, EEG, and more recently, spiking neural networks. So neuroimaging, then we use broadly machine learning. So, basically Bayesian inference. That's one of the themes that we have, and it all through the modeling being done using dynamical systems. So this is sort of three corners. And what are the levels of abstractions? So we are developing theory, both conceptually and also mathematical theories. And then we operationalize them using developing algorithms, for example, DCM. And we also do experiments. So and that's something new to our lab. So I'll talk about it a bit later on. So this is what we are doing. Let me now get into the main topic today, which is on brain connectivity. And I guess lots of people here are very familiar with these ideas, so just quickly. And this is again a sort of demarcation between these different sort of connectivity, which is not very exhaustive, but here we go. We have a structural connectivity, which refers to connectivity, which is measured by using diffusion MRI and probabilistic spectrography methods. But these spectrography methods and with diffusion imaging, it is as probably I should get my pointer to. Okay, so with structural connectivity, it is not possible to look at the directional direction of those white matter tracks and how the information is flowing on them. So these are three brain regions, and inherently these are undirected This is undirected connectivity, although we have done some work with Andrew Zaleski's group, led by Kyle Zepin, who used communication asymmetry in those white matter pathways to look at asymmetric communication. But it's more post hoc rather than inherently these imaging methods are undirected. Functional connectivity, which defined as the statistical dependencies of correlations. So by definition, the correlations are also undirected. So they only tell you about that activity in one brain region is correlated with another one. Okay, so the focus of today's talk would be on effective connectivity. Effective connectivity is defined as causal interactions between brain regions. And I will talk a lot more about this and how we actually define causality here, which has been something of ongoing interest and debates, that how do we define causality in DCM, for example. It's sort of a loaded word, so we will talk about it a bit more. Okay, and my talk would also, today I will also talk a lot more about the relationship between functional and effective connectivity. Okay, so here I will just start with this, with the problem definition, so to speak. So a connectivity reconstruction problem. So this is basically something which is done in silico simulations. So what I have here is three different brain regions which are connected, and there are connectivity weights on them. And we use this structure to run our forward model. And I'll talk about what forward model is being used, but what we do is we run our generated with a forward model, and we get time series. So these are the three time series that we generate from these three brain regions, depending on how they're interacting with each other. So you could see that if I use these three time series, I could calculate the correlations, the functional connectivity, and I get something like 0.77, 0.79, 0.49. What I'm trying to show here is this, that we can just change some of those connectivity weights here while the structure remains the same. They're all bidirectionally connected brain regions. What we see is this, that this connectivity, I shouldn't say connectivity, the functional connectivity here, which is 0.79, or the correlation goes from 0.79 to 0.18. We further change this, and for example, this connectivity, which is or functional connectivity, which is 0.77, converts into a negative correlation of 0.87 or minus 0.87. Similarly, we can change those weights again, and what you will observe is that these brain regions are not at all correlated. Yeah. So basically, what I'm trying to show is this, that this although the structure remains the same, how the interactions happen between these brain regions, they actually can give you very different observations that you are, and they give you very different inference that you can, or the interpretation that you can do from those observations. Now, as you could see, this is a mathematically, this is an ill-posed problem. So those who are working in engineering, they know about these mathematical problems. They exist in many different domains, not only in neuroscience or neuroimaging, as I'm trying to motivate here, many different areas where they are latent spaces or latent causes, and there are observations, and the mapping between these are always ill-posed or many-to-one mappings. Now, how do we actually now try to solve this problem? And this is really done by regularization. And sorry, so I'll talk a bit more about this now. And what I'm proposing here is this, that these interactions there we define as those causes or the effective connectivity. And this is where the name causal modeling is coming, where the causes are just basically causal architectures here, rather than counterfactual causality that's being discussed in statistics. And these observations or the consequences are the functional connectivity or the correlations that we can actually observe. So these are hidden causes and these are the observed consequences. And this mapping between them is ill-posed or regenerate mapping. So once I have this problem here, I can then say, okay, what we can do here? What we can do is this: we can now write down our generative model as a way to start, as a starting point. So what I have here is a forward dynamic causal model or the forward generative model. So what we have here is we start here with some endogenous fluctuations, or they could be basically a stimulus. So it could be deterministic stimulus, for example, an fMRI experiment where you have auditory or visual input. It could be a boxcar function which enters the sensory input which goes in, and it perturbs this dynamical system. Here I'm modeling brain dynamics as a first order differential equation where X are the current brain states, and X dot here is the first order differential equation, which is function of current states, some inputs, and the connectivity. How these brain regions are the causal architecture that I was talking about, which is shown here. This could be resting state that I will be mostly talking about today. So these could be endogenous fluctuations in the absence of an exogenous input. So this enters, this changes this system of equations, or which we mimic as modeling the brain function, and we get something out, which is the observed signal Y, or the bold signal, which is basically some observation function. In the case of fMRI, this G is a hemodynamic model, and I'll talk about a bit more later on. It could be EEG or MEG, where it could be a lead field, and some machine noise here, or the observation noise E. And from this, from Y, we can calculate functional connectivity. For example, this correlation or covariance matrix that I'm showing here, which is our data. Yeah. So this is from causes to consequences, or effective connectivity to functional connectivity. So I think for today's talk, the main idea is about there's a forward model that we can write down, and this is useful at many fronts. And everything else that I'll talk about will be, I think this is the main crux of the talk today. In practice, what happens is that these arrows on the other way, right? So what we can measure is functional connectivity, or the bold signal, right? And what we want is to find out what actually caused them, what was actually happening in the brain, those causal interactions that we can infer, but because we can't directly access them, so they are the latent causes. And what we could do is that once we have the forward model, we can use our observations and fit this data or some features of functional connectivity, let's say a covariance matrix or quasi-spectral densities, whatever. I mean, we can calculate all of these from our observed bold signal and use these as data feature, which is the functional connectivity, let's say, and then try to fit our data and try to infer the latent causes or the effective connectivity. Yeah. So here, as you see, this process of fitting is we will be working with within a Bayesian framework, and this is called Bayesian model inversion or Bayesian model estimation. While we do this, what do we want? What do we get at the end? The posterior densities over. So these are the connectivity parameters, their probability distributions that we are getting out, given the data sigma, which is a covariance matrix, let's say, or something which is generated from Y, or the data given some model. Yeah. What we are trying to do, basically, this is what we have done is that we have turned this whole thing into an optimization problem, and we are optimizing something here, what we call a model evidence. So this is, I have written a log model evidence. This is something which will be optimizing while we are fitting. This is what we're trying to minimize. Usually model evidence is not directly able, we are not usually able to numerically calculate this because it is, if you can imagine, number of parameters are pretty huge, and these requires integration over very high dimensional functionals. So what we usually do is we calculate a lower bound on this, but lots of people call it the elbow bound or the free energy. So this is basically what we are doing. But as you can imagine, as I was saying, that there can be this mapping from functional connectivity. The effective connectivity is not one-to-one mapping. It's not a unique mapping, right? So there could be many different combinations of those effective connectivity that can give you the same observations, yeah? And this is the ill-posedness of the problem, or the mapping between those causes to consequences. If I want to go from what I have observed and try to see what could be the most plausible cause of those observations of functional connectivity. So what I'm trying to show you is there are many different combinations. If I give you a simple example, for example, if I have an observation that I have some number 10, right? So number 10, and I tell you that, okay, it comes from some latent addition of 3 different numbers. For example, so number 10 could be coming from those addition of those 3 things, which are the latent things that we don't know. That it could be 5 plus 5 plus 0, it could be 4 plus 3 plus 3 plus, so it could be 5 plus 2 plus 3. So there are multiple additions that will give you always a number 10, right? And these are all possibilities that give you the same observation. And the idea is this, that what would be the most plausible thing that happened, which we can't directly measure, but we are trying to infer that. As I said, that with every possible, so we can define these as all possible models, and every model will have a score attached to them. This is what we call a model evidence. So every model or every cause will have a model evidence to them, right? And then what we could do is this, that once we have fit all those models, we could say that, okay, some of the models, so these are the log model evidence, and then posterior probabilities of model of all these models. And we see here, okay, there's one model, for example, which has the highest posterior probability or higher model evidence. And then we say, okay, this is the one that's basically is the most probable cause of what we observe. Yeah. So this could be a very sort of a hypothetical situation. Mostly what happens is this, that you don't have a single winning model. What you have is many different models which are very similar. So and what we do is we do a Bayesian model average, and then which is a weighted average of many different models, where the weighting of those models is based on how much evidence they have. So a model which has higher evidence will be contributing more average to that Bayesian model average. Anyway, so this is more conceptual, and this is how we operationalize dynamic causal modeling. One would then argue that, okay, there could be, if my model space is huge, there could be thousands or millions, or even sometimes hundreds of millions of possible models, and it becomes very difficult, right? How do we get these plausible model spaces? So there are multiple ways of dealing with this problem. That how do you actually define the models? Yeah. One is this, that either you handpick, so you use prior knowledge depending on what hypothesis and what problem you're solving, you're looking into a certain brain circuitry, and then you use previous literature and see, okay, what are the most plausible models that I would like to test? Yeah, so there could be some hand-picked 100, 200, 300 models that you think are the most plausible based on the existing data, and then you do the model comparison. So that's one thing. One way of doing that. It's basically based on prior knowledge in the area that the experimenter has. Yeah. Another way of doing is this, that we say that we don't know. We don't know what would be the possible model space, and we say that we want to do an exhaustive search, for example. So what we have is here is something what we call Bayesian model reduction. So Bayesian model reduction is it's not really new now. It's been used for years, which is a technology which lets you a very fast model scoring. So what you need to do is this, that in Bayesian model reduction is to only invert once the fully connected model. So let's say you have a model with 6 or 8 or 10 or whatever brain regions you want to look into, and you say that, okay, what I do is I go with a fully connected model where every brain region is connected to every brain region and all the connections are bidirectional. Yeah, I invert that model once. Yeah, and then what I do is I use this Bayesian model reduction technology, which lets me calculate the model, the model evidence of every nested model within that fully connected model by using a way of doing it is based on Savage Dickey density ratio. So what I'm saying is this, that under the assumption that the likelihood of any data under a model, under a given reduced model, is same as the likelihood of the data under a fully connected model. Yeah, given the reduced parameters are zero. So this is basically a very handy way of scoring a large number of models very quickly without requiring the inversion of every model within the fully connected model. So what you do is you do an inversion of a fully connected model, and then you let the Bayesian model reduction does the model scoring without inverting every model in the model space, and with this you can actually score millions of models within minutes. Yeah, it's approximated. It's a greedy search basically, and it is an approximation in a sense that it comes under that everything is Gaussian. Yeah, so there are of course some assumptions here, but it's a useful technique that we find very helpful in practice. With this, I would also talk a bit more about Bayesian model comparison and Bayesian model reduction. So model evidence or the free energy can actually be decomposed into two terms. So this is basically the first term, which shows the accuracy of the model fitting and also the complexity. That how many, so you could have very complex models, very big models with lots of parameters, and they would fit the data pretty well. But then, of course, the model complexity would be high. So when we are actually optimizing our cost function, which is the free energy, which is basically an accuracy minus complexity term, when we are optimizing this, we are trying to find out the right balance between the accuracy and the model complexity. So what we want is a and this is basically true for lots of things, is this, that we want to find out the simplest explanation of our data, which is also accurate. Yeah, so it has the which is accurate but also simple model, so that it is more explainable model, right? A model which is not like a black box, like with deep neural networks, but here we are talking about models which are explainable, transparent, yeah, where we can draw mechanistic inference from those models. And this is what we are trying to achieve here. What I would like to emphasize here is this, that the model evidence is also the function of what sort of data you use. For example, for functional MRI, we can actually use very simple Taylor expansions of those functionals. For example, here a bilinear approximation. So it's a very simple model. Here we have on the x-axis of this we have complexity, and on the y-axis we have the model evidence. And you see there's a sweet point here. While if we look at EEG data, and I'll talk a bit more about later on, which is that EEG can be we usually use very complex neural mass models with a huge number of model parameters. And at that point, you see that the complexity of those models is huge. And then you have another sweet point where you have the model evidence here. So what I'm trying to say is this, that model evidence is basically a function of what sort of models or data you're trying to explain. Yeah. Okay, so with that, this is a sort of a taxonomy of various different functional connectivity or brain connectivity measures which are in use. For example, cost covariance and cost spectral densities and cost correlation functions, coherence, causality. So what I'm trying to show here is this, that these are all these last two rows are second order statistics, and they can be derived in certain ways from state space models. So the main idea here is this, that if we want to infer the connectivity, we have to have a state space model. For example, the DCM, which is basically a state space model. Causality, where a lot of work recently has been done, where they can also be cast as a state space models. And once you have a state space model there, you can actually drive anything that you want, either with accurately or with some approximations, either in time domain or in frequency domain. So these ones in the middle, they are frequency domain equivalent. These are time domain equivalent. And this is what I'm trying to show here is this, that this is a one-way street. You can only go from top to bottom. Yeah, so you can drive, if you start here, you can drive whatever you want once you know the inferred connectivity. However, if you start here at the bottom with some covariances or correlations, you can't go back unless you have a forward model, yeah, or a generative model. So that's the whole idea here in some, okay, so that's basically, just to summarize, of course, this taxonomy is again not exhaustive. It's just a few of the measures which being employed in functional neuroimaging I'm talking about. Okay, let's move on to some more recent work. Let me see how I'm doing with the time. Okay, so yeah. So let's see how much I can cover. Some recent developments. So this is basically something that we have been looking at, where we can do model fusion or Bayesian model fusion, which is basically the idea is this, that can we combine EEG and functional MRI. So functional MRI and EEG fusion has been the holy grail of in neuroimaging, because they have so much complementary characteristics. Functional MRI having higher spatial resolution, but it's slow in time, while EEG has not much of a spatial resolution, but is very fast signal. Yeah, it has more temporal dynamics that you can do with them. So lots of people have tried to do EEG FOI concurrent measurements, but it's again a very difficult thing to do experimentally, that you can do both an acquisition, which is the MRI and EEG together. That's why it has never picked up as such. But what we are proposing here is this, that you don't need to do EEG and FRI acquisition in parallel. Rather, you can do a fusion after they have been separately acquired. So you do your MRI experiment and you do your EEG experiment separately, either on the same people or in different populations, depending on what you want to do, what is your hypothesis, and try to then combine those gracefully using something that I'm trying to express here. So what we are saying is this, that so we have some observations here, which are the induced responses of functional MRI, and we have some observations which are coming from EEG. Yeah, what we know is this, that they should be coming from the same sort of causes or the interaction between brain regions. So if we can have a same sort of a neuronal state equation, something that's common to both of them. Yeah, usually we use different sort of dynamics or models for functional MRI and different sort of neural mass models for EEG. What we are proposing is this, that can we use a same canonical microcircuit that I'll talk about in the next slide, and then the only thing different between these two measurements is the hemodynamics for fMRI and lead field, for example, in EEG. And once you have this, you can then combine these, because you have the same underlying model, which is same across both modalities. Yeah. Okay. So what then what is that causal model or a neural state equation model that we can propose that could be common to this, common to both modalities, and this is what we call a canonical microcircuit. So basically, this is I will try to explain what it is. So basically, this schematic is summarizing the generative model for each region or a node. So this model comprises of two sets of differential equations, one here, and then one set of hemodynamic differential equations here. Yeah. These are coupled via linear mapping, such that the neural states provides an input. Neural states provide an input to the hemodynamics here. Yeah. So if I try to explain this, the experimental input perturbs this neural circuitry. So there is an experimental input which perturbs this neural dynamics. The neural dynamics, which are modeled as a canonical microcircuit. This microcircuit comprises of 4 neural populations here. So you could see the sterate cells, superficial pyramidal cells, inhibitory interneurons, and deep pyramidal cells. Yeah, so these are the 4 different neural populations. Each population is equipped with 2 hidden states whose dynamics are described by a second order differential equation, as you could see here. Yeah, so these equations model the depolarization of each population in response to an experimental input and afferents from other populations. Yeah, as described here. So the 4 populations are coupled via intrinsic connections that correspond to known inter or intralaminar connectivity. The presynaptic activity at each population is then used to drive hemodynamic responses to the local field, local collaterals, innervating astrocytes whose end feed processes release vasodilatory signals. These signals can then enter a standard hemodynamic model to generate the bold signal. Yeah. And here in this graphic, the pink connections are inhibitory connections, the blue ones are excitatory, and green projections are mediating neurovascular coupling. So this is a description of this canonical microcircuit. And once you have that is common across both functional MRI and EEG, you can do something like this. Yeah. So what you do is this, that you go and acquire your EEG or MEG data. You first do inversion, yeah, using a canonical microcircuit. What you get is the posterior data, posteriors or the parameter posteriors. What you do is this, that you use these posteriors as priors for the functional MRI, which is again using the same canonical microcircuit, and do a second inversion of your fMRI data, and then you can infer the hemodynamics there. So this is basically what we call a Bayesian belief propagation, where you have 2 different sorts of data. You do first inversion using one of them, usually first one using EEG, You get your posteriors, put them as a prior and fMRI, and get your posteriors, and then this is the process that we, okay, so that's one sort of development that we have been working on. I will be a bit quicker, try to finish quickly. So this is again something lots of people are interested in whole brain DCMs, so something like what we had is a 36 node network, which is across larger scale networks like default mode, executive network, or dorsal attention, sensory motor. What we did was to develop a DCM which is very fast using spectral DCM. This is basically what it is doing is this, that it is writing the same sort of neural dynamics which are usually in time domain, writing the equivalent in frequency domain. And that's where the name comes, the spectral DCM. And we compared time domain DCM, which is what we call a stochastic DCM versus a spectral DCM. And we show that they are largely consistent. These are the free energy or log model evidence plots for every mode of these DCMs. So basically what we are saying that they give you the peak at the same, let's say, when we use 10 modes for stochastic or also for spectral DCM. What you get from these are these sort of causal graphs. These are different from usual functional connectivity graphs because they are directed. These nodes ages are both inhibitory and excitatory. So they give you more neurobiological plausibility, so to speak, by giving you the directionality and also the valence of the connections. Okay, so I have more on, but I think these couple of slides will probably require me a different talk where we use renormalization group, which is coming for statistical physics, looking at scales, different scales of dynamics and combining Markov blankets. I will just skip this. What I'm just trying to say here is that we have some multi-scale solutions which can be scaled the inference to thousands of brain regions. Something that we will probably talk about later in another talk at another equation. So I have a quick one where I just wanted to show a quick few slides where I could talk about application of those models that I was showing you to some of the work we are doing in psychedelics. But I think that I've already took about 40 minutes. So let me know if probably I need 5-6 minutes if it's
that's
okay with you.
Fine. Yeah, perfectly fine.
Yeah. Okay. Yeah. So I'll try to be brief and quick with this. So this is a new interest in our group where we are using some of those dynamic causal models to look at neural mechanisms of psychedelic induced altered states of consciousness. So just to give you, so my interest is coming from in psychedelics as a tool that they allow me to look at altered states of consciousness. Yeah. So psychedelics here as a way to understand, the neural basis of consciousness. While psychedelics are, being now sort of seen as the next big thing in psychiatry because of the huge potential to cure many different psychiatric disorders like depression, anxiety, and other things, which is hugely exciting area of work. But our interest mostly stems from using these as a tool to look at altered states of consciousness, to understand what are the neural bases for them. Yeah. For this, we use imaging, and I'll talk a bit more about that within a larger framework of predictive coding and predictive processing and an active inference. Some of the inference is done using dynamic causal modeling. For psychedelics, some of you who know a bit more about these psychoactive compounds, very important is the context in which they are given. For example, the setting, in what environment. It has to be very supportive, comfortable environment they are given, and also the mindset. How do we prepare people for the psychedelic experience beforehand is also an important thing. Anyway, so what I'm trying to say here on this slide quickly is this, that the harm potential of these psychoactive drugs, for example, the LSD or the magic mushrooms, which is the psilocybin, which is the active component in the magic mushrooms, is this, that the harm potential to both the users and others is very, very less when it's compared with things that are very much accepted, like alcohol. However, I should say that these data are coming from experiments which are done in lab in a highly controlled environment. So I'm not at all saying that these, they could be, they are definitely harmful possibilities if they are taken in an unsafe environment. Yeah. So this is data when these drugs are used in a safe, comfortable environment under the care of a psychotherapist. Yeah. Okay. So this is a very telling, sort of a schematic which shows that they are basically harmless when compared to other things like cocaine and tobacco and ice and all that. So, okay, so one thing here is this, that I will talk a bit more about subjective effects of these drugs before I go into the neural mechanisms very quickly. Quantifying the altered states of consciousness was initially problematic in early years of hallucinogenic research, but today we have very much of a validated instruments for assessing various aspects of consciousness using 5-dimensional altered states of consciousness instrument, which is 5-dimensional. It has, for example, things like oceanic boundlessness, anxious ego dissolution, vigilance, and visionary restructuralization, and so on. So there are 5 dimensions, and then all of these have sub-dimensions. For example, here we have under oceanic boundlessness, you could see something like experience of unity, blissful state, and so on. So what I'm trying to show is this, again, a meta-analysis where these things, these white squares are this placebo, and these are LSD. And you could see that there's a large effect size, for example, for oceanic boundlessness or visionary restructuralization, which are basically driven from blissful states, for example, and so on. So what I'm trying to say here is this, that these drugs do, there's now validated instruments which can quantify these altered states of consciousness using LSD, or in the same way using, for example, psilocybin. Quickly, I will just show you a quick example from our work from a couple of years ago, where we use LSD and looked at what are the neural mechanisms. So basically, what we are trying to say, we did was to test something what we call a thalamic filter model. Thalamic filter model talks about that the thalamus is a sort of a gatekeeper. So whatever sensory information that comes through your senses, like with your eyes, for example, this visual information passes through thalamus, and thalamus is like it's like a goalkeeper, a filter. So it lets only a fraction of information to the cortex, where it is usually then integrated and processed. But this thalamus is sort of a blocking most of the information and only passing some of it to the cortex. Yeah, what happens on the psychedelics is this, that and this is what the hypothesis was, is that this thalamus, this filter opens and it lets way more information into the cortex, and this results in the information overload, and we start to have all those psychedelic experiences. And this is where we use dynamic causal modeling. For example, we use 4 different brain regions, including thalamus and ventral striatum and posterior cingulate cortex and temporal areas. And what we showed was that the connectivity from thalamus to ventral striatum was increased, while the other way around, the connectivity from ventral striatum and temporal lobes to thalamus was decreased. So this is basically what we are talking about in terms of there was increased bottom-up connectivity compared to lowered top-down connectivity from cortex to subcortical areas. So basically, this is what we showed as a proof of concept, that this is the thalamic filter model, that how the LSD is interfering, or the predictive processing that's going on, the top-down and bottom-up signaling, which is being altered with LSD in the system. There's a working hypothesis for which more evidence is required, but this is a working hypothesis that which just quickly is one of the last slides. I know I'm being bit late. So what we are just trying to say is this, that the increased connectivity between sensory areas and the rest of the brain. So there's a increased connectivity in sensory areas, which is and there's a decrease in connectivity between association areas, and this balance due to the reduction of thalamic filtering results in the psychedelic experience. So this is basically a working hypothesis, and we are collecting more data. For example, in this one, this is a clinical trial which is happening currently in our lab. This is Australia's first clinical trial where psilocybin will be given to healthy people at an Australian campus. So this is what we are doing currently. The recruitment is ongoing. There's 60 people in this. There are 2 groups, meditators and non-meditators. Important here is this the context that I was talking about earlier, where how do we provide meditation and music, which are extremely important for a good psychedelic experience. So it's a multimodal imaging. We have both functional MRI and EEG. It's a longitudinal design with a baseline without drug, and the dosing day where we will be measuring MRI and EEG during the peak effects of psilocybin. There are lots of longitudinal behavioral measures. It's an ongoing work we are very close to start imaging, hopefully early into the next year. So with that, I would like to thank lots of people. So especially the computational neuroscience lab members, lots of people at UCL Methods, where I was based at for about 6 years at UCL. Lots of people at Monash with I collaborate, and many of our international collaborators and funders, and especially Carl Friston, who's been my long-time collaborator. Thank you.
Thank you very much. Thank you very much, Adeel.